Hello everyone, welcome to the webinar about next generation sequencing brought to you by Medical Design and Outsourcing Magazine and Ximedica. We would like to thank our presenters, Martha Najib and Rene Robert, for being here today. I'm Danielle Kirsch, Assistant Editor, and I'll be your moderator today. Just a couple of housekeeping details before we get started. You'll see several boxes on your desktop, all of which can be moved around to suit your preferences. Another box to note is the additional resources box. This box has websites and other information on this subject. Martha Najib is VP of Business Development and Rene Robert is Director of Engineering for Diagnostics. And without further ado, I will hand the presentation over.
Thanks for joining us. We're here today to talk about NGS-based diagnostics and opportunities within there to innovate healthcare solutions. My name is Rene Robert. I'm the Director of Engineering for Diagnostics here at Ximedica. I've been with the company for over 13 years. A history of capital systems, minimally invasive surgery devices, all different types of diagnostics platforms from point-of-care systems to hematology systems to sample prep devices.
Yeah, hi, my name is Martha Najib, and I'm Vice President of Business Development here at Ximedica. I have over 15 years of experience in molecular diagnostics with sales, marketing, product development, and global strategy and commercialization with Siemens, Abbott, Roche, and Cepheid.
We're going to start with a quick overview of Ximedica itself, and then we'll get into the NGS portion of it. Ximedica is a healthcare solutions delivery company. Our mission is to improve healthcare by delivering our clients innovative technologies to the market faster and more reliably than anybody else. We do this through complete integration of our product development processes, our quality systems, our expert resources, and our experiences in wide ranges of applications. We have over 200 staffed employees located in regional offices all across the U.S. and in Asia. Within our healthcare delivery solutions, we have a strong focus on IVD and life science systems. Those can be anything from diagnostic instrumentation, so hematology tests, immunoassay devices, up through molecular diagnostics, NGS systems, and then into consumables, consumable management, reagent management, and other single-use products. Within Ximedica, we've got a deep bench of expertise in all areas of instrument development, everything from control systems to thermal and fluid simulations to molecular biology to low and high volume manufacturing solutions. Essentially, anything you would need to bring an instrument from concept through to production. When working with our clients, we're more than a service provider. We're a trusted partner. We work diligently with our clients to understand their technologies and how those technologies best fit within their clinical application. Taking into consideration goals, timelines, and risks, we really work with our clients to develop the best strategy to maximize the benefit to them and the end user. At the end of the day, Ximedica is only successful when our clients are successful.
So the presentation today includes introduction to sequencing technology. We'll discuss NGS as a new diagnostic platform, and then review some opportunities for NGS innovation. There are three general approaches to clinical diagnostics. First, anatomical pathology, which visually analyzes tissues and cells. Chemistry assays that detect an analyte in a patient's sample. So, for example, PSA levels in blood for prostate cancer. For this presentation, we'll be focused on molecular diagnostics, which targets a patient's DNA and RNA to identify pathogens and disease states. So if we look at multiplex testing for cancer diagnostics, we'll use lung adenocarcinoma as an example. The number one killer is different mutations in one gene. The biomarker profile chart on the left demonstrates that there's not just one cause of lung cancer, there are hundreds. Many targeted therapies and clinical trials are available. However, if you look at the frequency of driver mutations, there are numerous genes to test and multiple mutations per gene. The issue is now you have a small amount of tissue and the guidelines require a less than 2-week testing turnaround. So the issue with complex diseases is that they may require reflex testing. Traditional molecular systems that utilize qPCR are great if you know what you're looking for, as they usually test 1 to 4 mutations per run. So is this single mutation present or not? The area of genomic interrogation is small, as little as less than 10 base pairs, and there are a few number of targets. So most qPCR instruments can handle about 4 to 5 differently colored probes per well, but they require multiple rounds of reflex testing. In the case of lung adenocarcinoma, you would need to perform multiple rounds of reflex testing, which results in sample depletion and extended time to results. Next generation sequencing delivers fast and comprehensive mutation testing. NGS is valuable in the case of lung adenocarcinoma. NGS allows you to test for more than a handful of small mutations and a long-tailed distribution of the types of mutations that you are researching. It covers tens of genes and thousands of mutations and results in less time. Most importantly, with limited tissue, you're not exhausting the sample and you can provide more rapid results in less than a week. This is the reason NGS has been adopted for these complex diseases. NGS shows great promise as a diagnostic tool for oncology and liquid biopsy, genetic testing and infectious disease testing. NGS surveillance adds to surveillance by identifying new and emerging strains. So there are 4 main sectors in the NGS process. The first is sample prep or sample extraction, and then you move to library preparation, DNA sequencing, and then data analysis. So let's start reviewing sample extraction. Sample types commonly used in molecular diagnostics are FNA, fine needle aspirates for thyroid cancer, FFPE, which is formalin-fixed, paraffin embedded for solid tumors and sarcomas, liquid biopsy for blood cancers like leukemia, inherited diseases. Genetic testing includes buccal swabs for inherited diseases like carrier screening or prenatal testing, blood spots, which are usually a prick in the heel for newborn screening. And then, of course, infectious disease is generally blood, plasma. You have cytology via either ThinPrep or SurePath, and urine for some sexual health testing, as well as swabs and culture. So when you take a look at sample extraction, some key considerations are obviously the user profile and environment. So you want to make sure the sample extraction kit and instrument works on the desired sample type. And in a clinical setting, there may be a preference to use an IVD-cleared instrument. Some customer drivers for sample prep are sample type compatibility and instrument flexibility. Throughput is a big deciding factor with batch size and linear time to process the samples. Hands-on time, IVD approval, and then, of course, downstream compatibility with multiple molecular diagnostic platforms. The takeaway here is really there is nothing about sample extraction is specific to NGS. So we talked about sample extraction. The next step in the NGS process is library prep. Library preparation is fairly conserved. Genomic DNA or RNA is extracted from the tissue. You fragment the genomic DNA, which means you're making it smaller. Once fragmented, polishing prepares the ends of the DNA for efficient adapter ligation. So end repair happens and poly-A tailing for the ligate adapters. The adapters then bind to the flow cell and enable the DNA fragment to be identified via the size selection, which will match the sequencing profile. Target enrichment specifically amplifies your region of interest, and then you have cleanup steps, normalization, and then you'll load onto the sequencer. Currently, library preparation is a very labor-intensive workflow. Companies are trying to make this easier with automation and reagent formatting. Instruments and materials used during NGS library prep include liquid handling or pipettes, beads and magnets for the bead separation, thermocyclers, a qPCR machine, which is used to normalize library concentrations, fluorometers and UV spectrometers, which measure DNA concentration, and fragment analyzers, which are used to measure the length or quality of the and concentration of the DNA and RNA fragments. These still take 1 to 2 days to do a batch of 12 to 48 samples. Most approaches to automated NGS library prep require creation of protocols for specific assays on existing laboratory automation. This works well for a research lab, but it's not great in a clinical setting, especially not for the biggest market, which is small to medium labs. Following library prep is DNA sequencing. Established systems include Illumina and Ion Torrent. However, some emergent systems are PacBio, Oxford Nanopore, and Complete Genomics, amongst others. The last step in the NGS process is the data analysis. Currently, the bioinformatic pipeline for genomic data starts with primary on instrument, secondary is variant calling, interpretation, and then results via a clinical report. So how do we integrate this valuable clinical genomic data into an established healthcare information system? So some general needs include data storage. You're going to have large gigabytes of data that's generated. Data processing, you need some significant compute power to support the bioinformatic pipelines. There's raw sequencing files that are mapped against reference genomes and to each other, and lots of quality scores and statistics are needed to call mutations with high confidence. The clinical annotation, moving from variants to clinical reports, and of course, databases of information. There's longitudinal studies and digital health as the RNA-seq or the MRD. Key considerations are scalable, flexible storage and compute power, servers versus cloud, so CAPEX versus operating budgets, HIPAA and data security, and of course, applications. Although NGS is a new diagnostic platform, workflow improvements are following same evolution as established diagnostic platforms. So if you think about where clinical chemistry or even traditional molecular diagnostics is today, it's amazing how far it's come. We're now seeing fully automated sample prep, processing, assay reading, fully tracked chain of custody. You've got the LIMS and soon the EHR integration, random access, and we're looking at completely decentralized with testing scaling from point of care all the way to product scale automation. The next-gen sequencing diagnostic instrumentation will follow a fairly standard development path. It's the same process where you start with chemistry and you optimize your assays. And we are starting to see and will continue to witness automating the instruments, which have many benefits, including it reduces the price of the cost per reportable, and it drives adoption. It decreases variability, decreases failure rates, IVD approvals, decreased hands-on time, data integration, and of course, test medical device approval and faster sample processing. When you look at data integration, this includes, of course, LIMS, EMR, and then a genomic analysis and longitudinal data collection and analysis. Despite a few approved tests, it's still in infancy for NGS in a clinical setting. The usual path is assay reader, processing an actual sample, then to automated sample extraction, and fully automated from sample to answer. If you look at diagnostics and molecular diagnostics, we are now fully automated from sample to answer. In the case of NGS, the majority of solutions are still in the first stage of assay reader. So the Illumina MySeq DX acts like a sample reader. Ion Torrent and Ion Chef are for semi-automated sample processing. If you look at the evolution of sample throughputs, it starts with batch processing, moves to mixed batches, and eventually ends up at random access. So diagnostics and molecular diagnostics are now offering random access. NGS is still in the mixed batches stage. As it becomes adopted clinically, platforms will need to offer a random access feature. centralization begins in reference labs, but decentralization actually transitions to large hospitals, medium hospitals, point of care, and most exciting is the transition to wearables and at-home use. Diagnostics is currently into the wearables and the at-home use. Molecular diagnostics is in point of care. However, we are very close to being both in wearables and at home. But NGS is still primarily in reference labs. Data connectivity evolves from LIMS, EMR, genomic analysis to longitudinal data and analysis. Both diagnostics and molecular diagnostics are currently EMR and are quickly evolving to integrate with digital health applications. When we look at NGS, it's still in the genomic analysis stage. So if you look at the current stage, there are a lot of opportunities in NGS. For example, we're seeing companies now look at trying to automate and integrate sample extraction and library prep. There are some manual library prep protocols that we're looking at automating onto a system. With DNA sequencing, there's some emerging technologies that are coming with some special IP. And then, of course, data analysis, we're looking to integrate with LIMS, EMR, and then, of course, cloud-based. So if we look at the future of clinical genomics, it's actually very exciting. So first of all, we touched on this testing locations. So genomic testing now or will be decentralized all the way through clinics, physician offices, and ultimately to the home. Connected devices are going to be important. Sequencing data will be combined with other clinical and environmental data thanks to wearable devices. If you look at digital health databases, there'll be multiple types of clinical data that will be incorporated into the structured and accessible databases. Testing frequency will become routine as periodic assessment of gene expression identifies harbingers of the disease. Bigger data sets will also become routine as they do periodic assessment of the gene expression data, genotypes, and of course connected devices to help identify harbingers of the disease. Sample types, we're already seeing things moving from tissue to non-invasive liquid biopsies. And what's really exciting is the personalized wellness planning. Lifestyle, diet, and exercise recommendations are optimized at an individual level based on measurable outcomes that move beyond DNA genotype that will include wellness biomarkers and clinical data. Instrumentation, as we're seeing in general, will become smaller, fully automated, and random access devices. And then data analytics and disease prediction will start to enable artificial intelligence analysis and proactively identify pre-disease signatures. So these are three areas that Rene is going to talk to in innovating NGS platforms.
Thanks, Martha. So three areas that we see the most potential for innovation around NGS revolves in automation, so making smaller, more fully automated random access instruments to address today's workflow challenges. The transfer of testing locations, so genomic testing is decentralized to clinics, physician offices, and ultimately to the home. And finally, the incorporation of digital health. Multiple types of data are incorporated into structured, accessible databases. So some of the design drivers that we see when we're commonly looking at automation, in particular for sample prep and library prep, can be broken down in a couple different areas. One of the big things is automating the assay itself. So taking the benchtop process that currently exists and replacing components of it with more automation-friendly equivalents. One of the big common ones is an isolation. So maybe there's a centrifuge step currently in the benchtop. That's a pretty hard thing to automate. I mean, it can be done, but it often leads to a large increase in size in the instrument. So we look to replace those types of isolation techniques with things like magnetic beads. Another area is trying to have common reagent volumes for dispensing. So in your current benchtop assay, you may be using 3 microliters of one reagent and 10 microliters of another reagent. what we try to do is look at the reagents and the concentrations and try to adjust them to get the dispensing volumes as close to the same as possible. That can make reduce the complexity of the consumable as well as the fluid dispensing technique. Another area that's common to look into is reagents and the consumable management. So everything from where these things are stored in the environment, in the user workflow, from how they get the reagents out of their freezers and where the consumables are stored to the instrument, and then how waste is managed. Another thing that we look at is tracking. That's a big design driver, is how our clients want to track their consumables and prevent counterfeiting. Typically, some clients look at barcode readers or RFID chips or various technologies out there, but that's a key decision that needs to be made. And finally, another key driver is flexibility and scalability. So one of the important things to understand when you're trying to automate your assay is how robust your assay currently is. How likely is it to change in the future? That can be a big design driver for how much flexibility needs to be left into the automation equipment. Will the automation equipment need to be able to adjust the size or volume of different reagents, for instance, or will have to add in steps? Or is your assay locked down to the point where you can really optimize the instrumentation? And then finally is scalability or future proofing. If you're going to add different tests in the future, do you need to have additional capability within your instrument for future tests and assays? In testing locations, as these devices become decentralized and transfer from the clinics to the physician office and ultimately the home, these are some of the key areas that we look at from a design standpoint. First is system integration and packaging. When you're designing a point-of-care device versus a large-scale core lab type device, how the individual modules fit together and how they're packaged becomes very critical. So your systems engineering and your interface requirements all become extremely important to understand early on, because you're trying to fit those puzzle pieces together in a much tighter fashion. Another big design decision is on consumable versus durable. So what things are going to be on your consumable versus stored in the durable instrument itself? Are you going to have reagents stored on the instrument because you're using a large volume of them, which will also lead to a cheaper consumable, but the interface between the consumable and the durable will be much more complicated. There'll be more seals, there'll be more risk of contamination, both from patient and from PCR contamination. What the user and the user error, how that impacts the design. So typically a lot of these devices are going into uncontrolled work environments. You don't know where the device is going to be installed or how it's installed. You don't know if it's going to be a clean or dirty environment. Will the device need to be CLIA waived? Will an untrained user need to be able to run a diagnostic test? Another important driver is oftentimes these durables, because there's now more of them and they're smaller, you're trying to get equivalent performance you'd see in a large scale instrument out of lower cost components. So lower cost imaging, lower cost detectors, things of that nature, heating sources. You're trying to really optimize that durable for cost and packaging. And a lot of times the way to do that is to look into lower cost components. So becomes a big design driver trying to get the same assay performance out of them. And finally, service and maintenance. Again, these devices are going to be installed in areas that that will need to be serviced, whether it's through calibrations or replacement components. So the service strategy is another thing that's considered early on in the design cycle to determine really what the overall, or is a big driver for the overall durable design. And then finally, the incorporation of digital health. So it's very important to take a user-centered design and data strategy to really maximize your outcome. So the way we do that is first we listen to the customer, or the end user, excuse me. We understand who the end user is, how they're going to be using the device, and we really review the data architecture. Then we organize that data. We research the content around it, and we understand how the data is going to format, or understand the data formats and how it's going to flow from the user through the device and ultimate to its end data storage situation, data storage location. Then we standardize. We standardize all the user-centered aspects of it, and we standardize all the integration aspects of it. We aggregate and analyze the data. We collect it on and determine if it's going to be collected on site or in the cloud, and we present the results in dashboards. And then finally, we use all this to deliver insights, clinical insights, predictive models, and ultimately increase utilization and improved outcome for our devices. So we talked a lot about NGS and clinical genomics. We want to summarize here what NGS is. It's sample extraction, it's library prep, it's DNA sequencing, it's data analysis. And really the current challenges that are around mainstream adoption for NGS revolves around the user workflows, instrumentation automation, and data connectivity. Finally, to address these challenges addressing these challenges will lead to decentralization of NGS-based testing and long-term growth of NGS as a diagnostic platform. And with that, we'll take your questions. Here's a question in regards to quality systems. What is different in your approach to quality systems in design history file that gives Ximedica an edge for FDA submission? It's a great question. Here at Ximedica, we have a very robust quality system. We've launched devices ranging from molecular diagnostics to implantable systems to connected health devices. And the way that we have tuned our quality system to be flexible enough to meet all those different types of devices, as well as rigorous enough to not essentially forget things or to miss certain aspects that are going to be important for your design history file, is all done through our quality service agreement we have with each client. And that basically is a checklist that we go through. It essentially starts with everything that you could possibly need to launch the strictest medical device out there. And for each client in each particular product application, we go through that checklist and we essentially decide which components are going to be required for that need. The nice thing about that is it allows us to not overburden a particular product with excessive quality documents or steps that may not be necessary, but it also prevents us from forgetting certain aspects of it, so that once the development process is all done, you don't realize you're missing, a particular risk management document or some early user testing data. So essentially, it's done through experience, but really through our quality service agreement we have with our clients. Martha, this is a good question for you. Where do you think NGS-based diagnostics are going in oncology?
Well, for tumor testing, the idea of a universal companion diagnostics is very appealing to both pharmaceutical and diagnostic companies. Utilizing a single test for all tumor samples that could lead directly to targeted therapies and enrollment in clinical trials. Also, in the liquid biopsy testing is really exciting, especially the ability to move from initial diagnosis into disease monitoring or even pre-disease surveillance. If you compare treatment for HIV and HCV to where oncology is going, you see a couple of interesting things. First of all, antiretroviral treatment contains multiple drugs that inhibit the virus in different but very complementary ways. Inexpensive liquid biopsies are used routinely to monitor viral load as well as viral genotype and evolving mutations. The prophylactic drug cocktail is adjusted for each patient based on the viral genotype and mutations that are present. And the results are that the viral load is maintained at near undetectable levels, and patients have a much better outcomes. I would love to see oncology start to use NGS and liquid biopsies in the same way.
Okay, here's a question that just came in. Magnetic separation technologies can be challenging to automate in microfluidic devices. Can you speak to your experiences in designing systems that utilize magnetic particles? Yes, incorporating magnetic separation does have its challenges. one of the things, particularly with microfluidic devices and cartridges, that can be a challenge is you not only are incorporating the beads and the associated magnets, but you also have to coordinate that with your flow control. So where the beads are in the fluid stream, where they are relative to even the flow front as the fluid moves through the microfluidics channels, and timing that with the magnets and the magnet location can cause some challenges. Sometimes if you're running multiple arrays in parallel, you can actually get crosstalk between the magnets. So those are some of the challenges when it comes to magnetic isolation inside of a microfluidics cartridge. Here's a question around library prep and FDA submission. If manual library prep process is cleared to the FDA now, why would you want to automate it? Wouldn't that make the submission process more complex and slower? Martha, why don't you take this one?
Yeah, so I think as in parallel with the evolution of both diagnostics, clinical diagnostics and molecular diagnostics, everything is moving towards automation. So in looking at library prep, automating library prep, it's the same benefits. You're looking at reduced cost per test. You're looking at decreased hands-on time, decreased variability, all of those same advantages of automating the library prep process.
Here's another question regarding NGS automation. How far away do you think NGS automation really is in terms of timelines? Martha, you want to take this one?
Yeah, sure. That's a great question. So NGS automation is really sooner than you'd think. I mean, within the 3 to 5 years that it actually takes to build and launch an instrument, I expect that you'll see these offerings on the market.
Here's a question in regards to infectious disease. Thanks for the talk. I appreciate the example of lung cancer. What opportunities do you see for NGS in infectious disease?
Yeah, and Rene, I can take that one also. So really the monitoring of emerging pathogens, so such as respiratory viruses or antibiotic-resistant strains of bacteria.
Yep, and the last one. So last question is around contamination in NGS. So sample cross-contamination can be confounding for NGS, given the potential for patient mix-ups. How do you prevent contamination within your NGS devices? Well, that's an interesting question. There's actually two types of contamination. We have patient mix-ups, so you're mixing patient A with patient B. And then we also have contamination in regards to PCR and amplification. So within the same patient, you're actually getting DNA that's a DNA strand that is essentially in the wrong spot and then gets amplified accidentally. So for the patient mix-up, that's really comes to the workflow analysis and understanding the environment and the user and how the device is going to be interacting with those environments. And really trying to design a system and a consumable that mitigates to the lowest risk possible any type of patient mix-up. And we do that through observations, we do that through UFMEAs, we do that through early risk assessments. And then for contamination, PCR contamination, that's really done with the design of the consumable and the automation system itself. And, you know, we add things like seals, like foil seals or doors that close different wells down and with spacing. So we try to minimize any, just from location standpoint, where certain amplification wells are relative to other wells.
Thanks everyone for tuning into this webinar. This presentation will be emailed to everyone later today. Thank you everyone for attending this webinar from Medical Design and Outsourcing.